Hey guys, hope you had a good day on Monday. Sorry I missed you. I'm going to go over some vocabulary you need to understand for chapter 16, which is mostly about population genetics, which we had started at the end of last unit. And hopefully this is a lot of review, but just remember a population is a group of individuals of the same species who are living in the same geographical area and they share a common gene pool. So, what is a gene pool? Um, Gene pool, remember that picture of the warthogs going into the pool? It's basically all the genetic information carried by the members of the population. In other words, all the alleles that they have. A mutation is a change in the DNA, and it's on the gene level, and it's something that's heritable, inherited. Gene flow is the movement of alleles between populations, and this happens through interbreeding. And natural selection, as we learned before, is just that idea of differential survival and reproduction of organisms as a result of an increase in the frequency of the best adapted traits. Non-random mating is where individuals are artificially selected. Here's a picture of a cow, bull, who has been artificially selected to have very big muscles. Kind of freaky, right? So it says this stud bull is the product of generations of selective breeding that have resulted in a double-muscled freak who can hardly walk. Populations and gene pools are affected by different factors, and they change over time. There's microevolution and there's macroevolution, and big difference is the word micro versus macro. And we all know that micro means small and macro means big. So microevolution is a change that happens within a species over dozens or hundreds of generations. So maybe this bird here that's smaller and lighter over time looks more different than this darker and larger one. Whereas macroevolution usually involves a much longer period of time and includes the origin of a new species. So over here again, they show this lizard changing colors, and that would be microevolution, but this lizard turning into a bird would be macroevolution. And a gene pool, again, is just that idea that there's a collection of alleles available among all the reproductive members in a population. So evolution occurs always on the population level. And we went over this definition before. And because individuals of a population can interbreed, they share common genes. But if they can no longer interbreed, then they won't share common genes. So each gene pool contains all the alleles for all the traits in that population. And here's an example right here. This is a Kermode bear, also known as a ghost bear. And these are white black bears. They're not albinos, and they live amongst black bears. And you'd wonder, like, why would a Kermode bear work or be selected for? And this article right here talks about how it turns out that the white spirit bears, when they are fishing above an area of moving water, their coat actually blends in with the waterfall there. And what are they looking for in that waterfall? Salmon. So the white bears, although they can be seen in the forest more easily, the salmon don't see them as easily. So that's kind of cool. So for evolution to happen in real populations, some of the gene frequencies must change over time. So not all the bears are turning white, but if eventually this became selected for, then maybe the white bears would win out. We know about the Hardy-Weinberg's law of equilibrium, and he had those 5 assumptions that if the population's large and mating is random, and there are no mutations, and there's no migration, and there's no natural selection, well then the allele frequency will stay the same. We know that these are really hard conditions to meet, and that allele frequencies in gene pools do change from one generation to the next. And this is evidence that evolution is happening. So we can use Hardy-Weinberg's equation as a baseline and then figure out if evolution is happening from there. So remember that the effect of natural selection can therefore be quantified. It's not just saying like, oh, the population changed. So if you change the allele frequency over time, you can assume evolution is happening. So here's a hypothetical gene pool. And I want you to stop the film for a second and count the frequency of big A's and little a's, and then figure out what the genotypic frequency is in this pool. Okay, figure it out. It turns out that there are 12 big A's in that pool, and there are 18 little a's. So the frequency of the big A plus the frequency of the little a's, 0.6 plus 0.4, and there's your solution for that problem. Now, check this out. What if, so as long as that condition is always met, as long as these guys continue mating, then the allele frequency will stay the same. But if the allele frequency changes, so now suppose that those guys that are on the sides jump into the pool and add a different allele frequency. So now instead of having 12 out of 30, we've got 12 out of 34 being big A's and 22 out of 34 being little a's. So the frequency of the recessive and dominant change, and you can see how that changes over time. So that showed you that Hardy-Weinberg's equilibrium wasn't maintained. There was a migration of swimmers or genes into the pool, and that changed the whole allele frequencies. So if migration were to stop and all of the other evolutions of evolution, like mutation, natural selection, non-random mating, didn't happen, then the population would then again stay at that new frequency. But it's important to note that a fifth factor affecting gene frequencies is always population size. So the larger a population is, the number of changes that occur by chance alone become kind of insignificant. So in the previous analogy, a small population was used just to simplify that. So why does it affect small populations more? Well, just like when you're flipping a coin, if you were to toss a coin 1,000 times and you got 750 heads, you'd be pretty surprised. But if you tossed a coin 4 times and got 3 heads, you'd be like, oh, no big deal. So chance has a greater influence upon gene frequencies in small populations. How much variation does a large population of sexually reproducing organisms have in their gene pool? Typically about 0.5% of their DNA bases, or A's, T's, C's, and G's in an organism are different. In fruit flies, they have 165 million base pairs, so that means that they've got 1 million nucleotide sites that differ. In humans, we've got 3 billion base pairs, so we've got about 15 million different or variable nucleotides. The mutation rate, in terms of how often we get new alleles, is one source of variation within a population, and due to all the times that we replicate DNA, the mutation rates of individual we have repair mechanisms. So it's actually not very common to have mutations, but because we have so many genes, there's always new mutations happening. They talked about the peppered moths in Crash Course. If you haven't seen that yet, you should. In 1850, it was really common. Light-colored moths lived on light-colored tree trunks, so it worked really well for them. They lived with this lichen, and everything worked well to be light-colored. If you were a dark moth on the light color, you didn't do so well. But in 1900, just 50 years later, check out this, the allele frequency flip-flopped, and that's because the light lichen died from pollution. And suddenly, if you were a light moth on a dark trunk, you stuck out like a sore thumb and you got eaten. So the dark tree trunk helped the dark moths do better. So light-colored moths became easy prey for the birds. And that's showing, a mutation is what allowed the dark ones to exist in the first place. Mutations can be good, bad, and neutral. They're random, and some are lethal and kill an individual before they're even born. Some are harmful, but masked by the dominant allele, so we don't even know they exist. Each of us carries about 7 or 8 lethal recessive genes. Some are neutral and have little or no effect on an organism's long-term survival. And some are actually beneficial, and they help the organism survive to reproductive age and reproduce. These are just showing, this is like a duck with a third leg. It doesn't really seem to hurt it right now, but here's a bear with an extra toe, and here's a sort of short-legged steer. So gene flow is just the exchange of alleles between populations, and this exchange happens through migration usually. Gene flow increases variation within a population and introduces new alleles. Genetic drift is the random change in a gene pool, and it happens when two isolated populations become more different or dissimilar. Some alleles get lost, others get fixed. So the cause of reduction in the frequency of heterozygotes. This causes a reduction in the frequency of the heterozygotes over time, and the smaller the population size, the faster it's seen. So when is genetic drift likely to happen in nature? Most populations are large enough so genetic drift doesn't happen, but some populations crash in numbers due to a natural disaster or over-harvesting, and you get something called the bottleneck effect. Other examples are when new organisms go to a habitat that they've never been at before and they start a new population. That's called the founder's effect. And that suddenly small population, just by chance, contains a different frequency of different genotypes than the original population. So the founder's effect happens when a few individuals migrate to a new place. So, and that small number of emigrants doesn't necessarily have the same allele structure or genetic structure as the main population. So they create a different sort of allele frequency. This happened with the Amish people in the 1700s. 200 German Amish immigrated to Pennsylvania to start a community, and those people carried a pretty high concentration of mutations for polydactylism. So individuals in this group tended to marry each other because there weren't that many of them, and there was a greater so there was a higher likelihood of having polydactylism within these people. Bottleneck effect is similar, where the population crashes and then comes back. Inbreeding is when there's breeding between close relatives, and the big problem with that is it causes a gradual increase in homozygosity. You lose the heterozygotes, and an example of this is that in most species, related individuals share about 80% of the same genes. Cheetahs, who were pretty much hunted to extinction, share about 99% of their genes because they were almost hunted to extinction, and then they started to come back. And this causes low survivorship. A lot of the animals are dying because of poor sperm quality, larger susceptibility to diseases. So the bottleneck effect is when an originally large population gets kind of crunched down to a small population, and the few survivors are likely to have a genetic structure that's unrepresentative of the ancestral population. So here's the bottle, only a few of the alleles come out or survive, and it's not representative of that original population. An example of that is the northern elephant seals. There's a northern population and a southern population. The northern ones were hunted almost to extinction. They were reduced to 20 individuals, and their population has rebounded to like 30,000, but they still have a lot less genetic variation than the population of southern elephant seals that wasn't so intensely hunted. So just comparing the two effects, they're pretty similar. Bottleneck effect is a sampling error as a result of only a few individuals surviving a population crash, and then the founder's effect is a sampling error as a result of movement of a few individuals away from the main population. I think I'm going to stop there. Just answer the questions up through founder's effect and bottleneck effect, and we'll talk about the rest in class on Wednesday. Thanks.